like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Welcome to the National University of Singapore Yong Lu Lin School of Medicine's Healthy Longevity webinar. Thanks for joining us. This is our first show of July. I'm coming to you from Zurich, but it's kind of an all Portugal night tonight, so except for me. So we'll be excited about that. Our speaker is Joao Pedro de Magalhaes. I know I got that wrong, but I tried. And he's going to be giving us a really interesting talk on longevity. I want to remind you to use the Q&A function to ask questions, and Diogo, also from Portugal, will get to those questions and ask them for you. Today, before we get to Joao Pedro, we're going to hear two presentations from younger speakers in the Healthy Longevity program. The first is from Sabrina Adam, who'll be telling us about some exciting research from her laboratory. The second is from Basil Kua, who'll be describing a recent paper on the role of TDP-43 in neurodegeneration.
Thank you for the introduction. As mentioned, my name is Sabrina. I'm a research assistant under Dr. Faiha Abdul Jalil. And today I will be introducing our lab, which is a tissue regeneration and repair lab, as well as some of the projects that we're venturing into. So our lab primarily works with cardiovascular and stem cells, and our research will be focusing on getting a deeper understanding on cardiovascular and stem cell aging, as well as using stem cells and all that we can produce and derive from them as a treatment to reverse or slow down the aging process. So what are stem cells? Stem cells function mainly to provide new cells for the body to grow, as well as to replace old and damaged cells. They can be defined by two very unique characteristics. One would be their ability to self-renew continuously. And secondly would be pluripotency, which is the ability to transform into many different types of cells. And a common misconception of stem cells that we have is that they are only found in bone marrows, but in fact they can be found in various other organs, such as the brain, liver, heart, even blood and fat cells. So there are many different ways that we can exploit the therapeutic potential of stem cells. So for example, one of our projects, we look into thalassemia disorder, which is an inherited genetic disorder affecting the capability of red blood cells to deliver oxygen adequately. And our project aims to utilize these reprogrammed stem cells, or iPSCs, standing for induced pluripotent stem cells, as a treatment for this disease. So we intend to firstly collect the buccal cells or oral cells of thalassemia patients and correcting this genetic mutation and finally transforming them into therapeutic cells before reintroducing them into the patients. So it is noteworthy to mention that the therapeutic potentials of stem cells goes beyond cellular therapy, such as the one I've mentioned previously. And stem cells may also be used as a factory per se to produce therapeutic biomaterials by collecting their secretions. So this secretion, it's called secretome. It comprises of two components, cytokines and extracellular vesicles, which contains materials like proteins and growth factors, enzymes, even genetic materials like DNA and mRNA. So The contents of the secretomes help to firstly protect the cells from further damage. It does this by mediating the inflammatory responses of the cells. And then secondly, it does promote regeneration so that you can repair the damages of the cells. We'll be applying the therapeutic potential of stem cell secretome into a study involving progeria patients. So progeria is a rare genetic disorder which patients suffer premature aging and often pass in their early teenage years due to cardiovascular diseases. In this study, we will introduce the secreted secretome collected from amniotic fluid stem cells, and we'll introduce them into the patient's heart cells. And we hypothesize that the secretome will mediate the inflammatory responses in the heart cells of progeria patients, so as to manage the acceleration of cardiovascular disease development in these patients. And lastly, I would like to mention one of the special machines that we have in our lab, which is the Luminex Machine. The machine employs this laser-based technology that detects specific biomaterials in samples. So for example, here we have some secretome that we played onto these magnetic customized beads that will attach the molecules found in the secretome. And as the machine evaluates the samples with the lasers, it generates some data for us that allows us to recognize the biomaterials in the secretome. In other simpler words, this machine allows us to detect and quantify biomaterials in a given sample. So that's all for today. Thank you so much for your interest and attention. I sincerely appreciate the opportunity to have presented. Special acknowledgements to Dr. Farhan and Dr. Marik, as well as BioRender for all the images that were created in the presentation. Thank you.
Hi everyone, as mentioned, my name is Basil, a graduate student from NUS School of Medicine. Did you know our brains shrink with increasing age, affecting our cognitive abilities such as learning and memory? Yet, up to today, we are merely scratching the surface in understanding the horrifying degenerative effects of aging. To facilitate the identification of treatment, we first need to understand how different cell types that make up our entire brain could contribute to the detrimental effects we often see in aging. And that is exactly what we are doing here in the Lab of Molecular Neurodegeneration, headed by Dr. Ling Suo Qian. So aging is an irreversible process that occurs in each and every one of us. As the brain ages, changes in the brain microenvironment starts to manifest. These changes are such as the presence of protein aggregates, dysfunctions of lysosome, as well as cellular senescence. Although aging is commonly known to be a major risk factor for neurodegenerative diseases, the exact mechanism has yet to be identified. In our lab, one of the key proteins of interest is TDP-43. Under normal conditions, TDP-43 is predominantly localized within the nucleus, where it binds to both DNA and RNA. In addition, it's also known to play a key role in the life cycle of RNA, where it participates in the synthesis, processing, as well as the transport of RNA. Although mainly found within the nucleus, TDP-43 is able to shuttle into the cytoplasm, where it participates in processes such as translation from RNA into proteins. However, in multiple neurodegenerative diseases such as amyotrophic lateral sclerosis and Alzheimer's disease, TDP-43 loses its predominance in the nucleus and starts accumulating within the cytoplasm. This shift in location results in the loss of its normal function. What complicates the issue even more would be the fact that this phenomenon happens in both neurons, which are the fundamental units of the brain, as well as the supporting cells of the brain known as the glial cells. For instance, around 97% of patients with ALS, as well as 36% of healthy age individuals were observed to have formations of TDP-43 aggregates in the cytoplasm. Due to the huge number of processes carried out by TDP-43, as well as the manifestations of aggregates in most cell types within the brain, it is unsurprising that dysfunction of TDP-43 may contribute to diseases. Previously, several labs along with ours have selectively deleted TDP-43 in motor neurons as well as subpopulation of glia to understand how loss of TDP-43 function could contribute to neurodegeneration. Collectively, these studies have highlighted that different cell types in the brain have different dependency on TDP-43. However, most of these studies were conducted within the spinal cord. Hence, we wanted to fill in the gap in knowledge in understanding the normal functions of TDP-43 in specific cells of the brain and how the loss of function could contribute to neurodegenerative diseases. To specifically delete the protein of interest in a cell type, we utilize a genetically engineered mouse model to generate cell-specific deletion of TDP-43. Subsequently, one of the key areas that we were interested in is known as the hippocampus, as it plays a major role in learning and memory, which is often affected in aging and neurodegeneration. By first conducting some cell biological studies such as immunohistochemistry, we seek to identify the visible changes that had occurred when TDP-43 is deleted. Ultimately, we noticed that the structures of the cells have drastically changed in the mutant compared to the control mice. Furthermore, the placement of the cells have been disrupted when TDP-43 is deleted. This, together with the fact that there was around 20% decrease in the volume of hippocampus in the mutant mice, suggests that TDP-43 potentially plays a monumental role in development of hippocampus or maintenance of stem cells. This could indirectly result in the decline in cognition as seen in aging. By elucidating the underlying role of TDP-43 dysfunction, we hope to expand our understanding and potentially facilitate the discovery of treatments for neurodegenerative disease to attain healthy aging. Thank you for your time. I hope you find this snippet of information interesting just as I did.
Dr. de Magalhaes graduated in 1999 from his hometown university in Porto, and then went on to get a PhD from the University of Namur in Belgium. Following a postdoc with George Church at Harvard Medical School, he joined the University of Liverpool in 2008 and has been there until recently when he decided to move to the University of Birmingham to lead the Genomics of Aging and Rejuvenation Lab. I'll let him tell you a little bit about his research, but he's also given a number of TEDx talks, and he's the CSO of Youth Biotherapeutics, which is looking at partial reprogramming. So there's a lot of things going on. It's great to have you on the show. The title of the talk is Solving Aging from Home with Computational Approaches. Thanks for joining us.
Thank you. Thank you very much, Brian, for the kind words and the invitation and introduction. So let me share my screen, and I'll tell you a little bit. I'll give you a brief overview of some projects we've been involved in recently and some particularly computational approaches. Can you see my slides?
Yes, we're good.
Okay, that's great. So yes, thank you again for the invitation and the opportunity to tell you a bit about our work. I'll start with a brief introduction, and then I'll mention some of the computational analysis we've been doing. I don't need, given the series focus on longevity, I don't need to convince you of the importance of research on aging and longevity. So I'll skip that. I'm also sure you're aware of the impact the pandemic had on our way of life and our way of working. So COVID-19 had a major impact, and I'll focus mostly on computational approaches and really how much we can do from home with computational approaches to advance our knowledge of aging, longevity, and interventions and potential therapies. And of course, COVID-19 is an age-related disease. As I'm sure you're aware as well, this is data from China from the start of the pandemic, but it's fairly similar across countries. And it really shows the exponential increase in mortality from COVID-19 with age. So it's clearly an age-related disease. Or to put it another way, something happens to our bodies that predisposes to a variety of diseases, including cancer, cardiovascular diseases, neurodegenerative diseases, and of course, many infectious diseases, COVID-19, but pneumonia, the common flu, of course, it's much more dangerous in elderly patients. So something happens. And clearly, we need to understand that, and ultimately, I would like to develop interventions for it. Now, unfortunately, we still don't understand why we age. I mean, we understand some of the changes, okay? So there's changes in the immune system, for example. So we understand some of the changes that occur with age, but we don't understand the causality of it. We don't understand what drives what molecular mechanisms, cellular mechanisms, physiological mechanisms are driving the process of aging, or which multiple mechanisms. But so we don't really understand the hierarchy of what is happening during aging, although we are aware of some changes that occur during aging. And that's a major problem. Now, there are theories of aging. I'm sure you're aware of many, the telomeres, DNA damage, oxidative damage. I guess I'm sure you're familiar with the hallmarks of aging. But my point is that we don't know if they are true or not. In fact, last year with David James, we published this criticism of the hallmarks of aging, that in the sense that they're interesting as and useful as an introduction, but they may be wrong from a mechanistic perspective. So they're hypotheses, and they may or may not be right. And same for the pillars of aging that Brian was involved in. I think they're good hypotheses, but they're not proven yet. And so we still need to do, I think, a lot of work in order to actually demonstrate that any of these mechanisms is really causal in aging. And they may be wrong. There's maybe something else that we're missing that is really the driver of aging, and we're just identifying passengers of aging instead of drivers of aging. So, I mean, with that said, there's been a lot of advances in aging. I'm sure you're aware of the great advances we've seen in terms of genetic and dietary manipulations of aging in model systems. So just very briefly, as an introduction, since H1 was identified in C. elegans by Tom Johnson and colleagues, there's been huge advances in the field from Cynthia Kenyon, Gary Ruvkun, Lenny Guarenti, and many others that really shown that the process of aging is surprisingly plastic. So we can manipulate individual genes, and this has a significant impact on the process of aging. So this is something I've always found fascinating. And actually, when I was a PhD student, I started this collection of databases and tools called the Human Aging Genomic Resources. And so one of the databases that I started, God, it's making me feel old, but it was about 20 years ago, was this GeneAge database of age-related genes. So it's a curated database in the sense that I look into the literature. Well, we have other people that contribute to it as well. And then we look at genes, individual genes, and when you genetically manipulate it, overexpression, mutation, silencing, then they have a significant impact on aging or longevity. And these databases have evolved into the benchmark databases in the field. They have hundreds of thousands of visitors, hundreds of citations. And so as a side note as well, we also have, I also maintain on Facebook and Twitter pages with the latest papers. So if there's an interesting paper, I just post it there. So if you want to keep up to pace with the latest biology of aging papers and news, I invite you to follow our Twitter or Facebook pages. Going back to GeneAge, so I think it's really a reflection of how much we've progressed in the field, which is quite remarkable, I think. So this is the latest version of GeneAge, and as you can see, there's over 2,000 genes in model systems that when we can genetically manipulate them, they have a significant impact on aging or longevity. So in worms alone, I mean, there's nearly 900 genes, over 900 in flies. I mean, there's single gene manipulations in worms. It's actually a different allele of H1 that increase lifespan nearly tenfold. So, which is quite remarkable that you tweak a single gene and it makes animals live instead of a few weeks for several months. So it's quite remarkable how much we've advanced. I mean, even in mice, which of course are more complex than worms, we have over 100 genes that impact on aging or longevity in mice. So genes that accelerate aging or retard aging in mice, and you have an increase in lifespan up to about 50%. So I think it's quite remarkable how much we've advanced in this, what I call the gerontome. It's fantastic that we have all of these manipulations of aging, that we can manipulate aging in model systems, in animal models. I think it's a huge breakthrough for the field. So in a way, I think of it from a more genetics perspective, which is, if you think about it, the genome, the human genome is 3 billion base pairs, 20,000 protein coding genes, God knows how many non-coding genes it has. But really, very small changes in the genome can have a significant impact on aging. You can have a very small tweak in the genome and accelerate aging or retard aging, at least in model systems. And there's no reason to think we can't do the same in humans. So it's quite remarkable how we have this digital code in the DNA that can, with very relatively subtle changes, can have a significant impact on aging and longevity. And that's something I always found fascinating. Ever since I started, well, even when I was an undergraduate, I thought it was fascinating how the information for life was encoded in the genome. And then later, when I started working on aging, how much it impacts on aging and longevity. So on the one hand, I think that's fascinating, but on the other hand, it also means that we need to try to understand the genome, try to decode the genome, try to understand that information. But there's a lot of genes, of course. As I mentioned, there's 2,000 genes in model systems that impact on aging. And of course, they interact with each other and with the environment in nonlinear ways. So really the challenge now is to understand complex systems as a whole. So I think not just in aging, but biomedical research and biology is becoming a mathematical problem in the sense that we are generating large amounts of data. And the shift really is from generating large amounts of data to analyzing data. And that really is the challenge now. And when you think about a complex process like aging that has hundreds, thousands of genes influencing it, really the challenging is to bring those quantitative approaches and those modeling and computational methods to really understand a complex phenotype from all of these hundreds of thousands of interacting parts. And so that's what that's one of the concepts that we've been focusing on, trying to understand these complex systems from individual components. So I guess our lab, we have various focus, and I don't have time to go into detail on all of them. But broadly speaking, of course, we have a focus on aging and genetics. We do some work on dietary manipulations that I'll briefly touch upon. We do some work on evolution, which I don't have time to go into detail today. We work with cell models, long-lived species. We focus on complex diseases as well, which I'll touch upon briefly today. And we do a lot of work on computational and systems approaches. And there's a lot of computational approaches in these various, understanding these various processes. So I'll tell you today, I'll tell you, very briefly about a few different projects that we've involved in recently in applying computational strategies. Again, things you can do from home. Applying computational methods and approaches to gather new insights, many of which, well, at least some of which, we then went on and test experimentally, either ourselves or with collaborators. So let me tell you about a couple of this and try to persuade you that really we need these computational approaches to make sense of all the data that we are generating. So I'll start with a project we did a couple of years back, and well, so I did my PhD actually in cell senescence. And then a few years back, we started focusing on cell senescence again, trying to understand it from a perspective of complex phenotype. So just as a brief introduction, so I tend to define senescent cells as cells that should be dividing, but are not dividing, have irreversibly stopped dividing. So that's how I tend to I tend to define senescent cells. Now, what seems to happen, at least in mice, is that senescent cells accumulate in normal tissues, either due to telomere shortening or stress-induced mechanisms, and then they disrupt tissue function. They also secrete inflammatory cytokines and contribute to inflammation. And there's a lot of interest in developing therapies like senolytics that ablate senescent cells specifically that can help with age-related diseases and with aging. There's various companies working on the topic, like Unity and many others. So it's a very active field of research. So that's cell senescence. So we've tried to bring in some of our computational and systems biology approaches to the problem of cell senescence. So we did various analyses, which I'll very briefly summarize in just a couple of slides. So basically, the first thing actually we did a few years back was we did this meta-analysis of transcriptomes of gene expression profiles of cell senescence. So these are young fibroblasts, by the way, and these are senescence fibroblasts. The photos I took about 20 years ago, or nearly 20 years ago when I was a PhD student, by the way. And as you can see, senescent cells, they're much more heterogeneous, some are long, big, and so the morphologically they're different. And then there's other markers of cell senescence you can use, although there's not one universal marker. But anyway, so in this multidimensional analysis of cell senescence, we started by doing a gene expression meta-analysis to identify signatures of cell senescence. And we look at, we identify these signatures with over 500 genes overexpressed in cell senescence and over 700 genes going down in cell senescence. So we found this signature of cell senescence. I mean, we did other things. We looked into the literature for gene manipulations, trying to infer if some of these genes are causal or not, if they are promoters or inhibitors of cell senescence. So we do other analysis I'm not going to go into detail. But basically, we had these groups of genes upregulated and downregulated for cell senescence. Then we did various network analysis. Now, one of the network types that we focus is co-expression networks. So co-expression, so basically for two pairs of genes, you can look in a variety of conditions, and you can look how strongly they correlate their expression with each other. And genes that tend to correlate very strongly with expression, they tend to have similar functions, or at least participate in similar pathways. And so from that, you can build networks based on their co-expression strength. And from those networks, you can get various insights. I mean, we did this big review, actually. It's a big paper a few years back from particularly Sipko Van Dam, who was a PhD student in our lab, who really drove this line of work in co-expression analysis. But you can do, very briefly, you can look at regulatory interactions. You can do functional enrichment analysis, and you can find new regulators of a particular process. And you can also use a guilt by association analysis to identify new candidates. So if you have a particular gene associated with a disease that interacts with lots of genes, some of those not yet studied in the context of that disease. I mean, you can do statistics behind it, but you can try to prioritize and identify new genes for studies based on their network properties and where they sit in a network. Aging, senescence, or disease-related genes. So to show you an example, we did this for cell senescence. And so just one very brief thing before showing that. So we actually developed, particularly Sipko and now others in the lab have developed a set of tools to do co-expression analysis. If you're interested, they're called Gene Friends. And basically what we do is we download all of the data we can get our hands on. We started with microarray, we use RNA-seq data. So it takes a long time to download the data. It's one of the bottlenecks of this analysis. And then we build a whole genome co-expression map. So we have 80,000 transcripts by 80,000 transcripts. And you can use this tool, for example, to infer functions of unstudied genes. So I said, if you have a gene that interacts with a lot of mitochondrial genes, chances are it plays a role in mitochondria. And you can use it to prioritize, as I mentioned, using a guilt by association method, to prioritize genes with associations with any gene with any disease or process, if you have a seed list of genes for that disease or process. So, I mean, we've published this a few years back. We have a preprint now that's currently submitted with a new version. But I invite you to look into our tools and download our data for doing your own co-expression analysis. So in cell senescence, we built a cell senescence co-expression network, which is what you see here. And as you can see, the genes from the network, they cluster into different groups. And each of these colors represents a particular function. I don't actually remember exactly the colors, but I know one of these clusters will be cell division, some other will be immune system responses, other will be DNA repair, and so on. So, they cluster into particular processes. Which makes sense because we know cell senescence involves all of these processes. But then what you can also do is you can use these network properties and guilt by association to identify candidate genes based on their position in the network. So here we have our seed list of genes and also their interaction partners. So we have genes that have not been associated with cell senescence yet, but we can look at the most probable, we can rank them based on how strongly they are positioned in a network. So to make a long story short, we looked at various network properties. I don't have time to go into details, but basically you do a ranking of the genes, and then together with collaborators, we tested this experimentally. So the experiments were actually done in London with Cleo Bishop at Queen's Mary University, which is what you see here. So here on the left, you see the 26 genes we used, plus a negative and a positive control. And you have various markers of senescence, from cell number, you have cell area, you have P16, P21, so traditional markers of senescence. And when you genetically manipulate these genes, this was done with RNAi. So when you silence the genes, you can see a lot of them, actually most of them have some impact on some biomarkers of senescence. So of the 26 genes tested, 6 of them were strong hits. That means when you silence them, they have a strong, well, they impact on multiple biomarkers of senescence. And there were 12 moderate hits that also impact, but to a lower degree, in biomarkers of senescence. So what we show here is that you can use this network biology, this computational analysis, to identify new genes associated with a given process, in this case, cellular senescence. And I mean, we've done this for aging, we've done it for longevity, which we tested in C. elegans, we've done this for different processes. But I thought this was quite remarkable because the hit ratio was so high. I mean, most of the genes we identified actually turns out they play, or they're associated with senescence. So that's one type of analysis we've done. So let me change gears a little bit and let's show you a different type of analysis. Okay. So the other kind of analysis we focus on is more on pharmacology or network pharmacology. So the idea is that just like you can get gene expression profiles, you can get molecular signatures of senescence, you can get molecular signatures of longevity and of aging. You can also get, actually, you can download gene expression molecular signatures of drugs. And so we started this project some years back. And the idea was very simple at the time, was just, hey, can we identify compounds, drugs, which profiles overlap with longevity? But we actually use caloric restriction. We actually use many things. The one that worked best was caloric restriction, gene expression profiles. I can go into more details later if you wish. But basically, we do this analysis, and then we basically got the gene expression profiles of longevity / caloric restriction, and then we look at over 1,000 drugs and we see which drugs come out. And we showed that actually the top drug was rapamycin, which I thought was very interesting, showed that we were on the right track. And then we actually did this in our lab. We actually went out and tested the 5 drugs, top drugs we identified in words, well, one of them being rapamycin, which was sort of a positive control. And of the 5 drugs we tested, 4 of them extended lifespan, actually in a caloric restriction manner. So we can show that this network pharmacology approach can reveal new compounds associated with longevity. So, for example, one of the new compounds was allantoin that I know Diogo, he's studying in Singapore, for example. And I mean, I won't go into it, but now we're actually looking into some of the mechanisms of these drugs. That's a collaboration with Colin Hewald in Zurich. But that's not something that'll be a topic for another talk. So that's one approach we've done. Now, a kind of related approach in terms of computational analysis is machine learning approaches. So, I mean, we did this review a few years back on applying machine learning to aging research. I tend to define machine learning. I think a good analogy is, you know, sometimes on Facebook or online you find this website that allows you to predict your Game of Thrones character. So you put in your characteristics, you know, you're male or female, do you like killing people, yes or no? And then what that algorithm does is that it matches your characteristics to the characteristics of Game of Thrones characters. So that's a machine learning algorithm, basically. And what we do is basically the same thing. Instead of using Game of Thrones characteristics, we use gene characteristics or drug characteristics to match which genes and which drugs have profiles or have characteristics that are similar to those you'd expect to be associated with longevity. So we did this, one of the first ones we did actually was on pharmacology, and this was done by Joao, who, as I mentioned, well, he's on the call, he's now in Singapore, where we use chemical and biological properties of compounds to predict new life-extending compounds using this machine learning approach. I mean, more recently, we've also been applying this to genes. I mean, particularly this is work with Alex Freitas, my long-term collaborator at University of Kent. We've been looking at genes associated with cognitive aging. So we look at properties of genes previously associated with cognitive aging and cognitive function, and then we try to identify new genes associated with it. And to make a long story short, we found about 10 genes, one of which, when we look at epidemiological data, in collaboration with Ian Deary in Edinburgh, one of them is a potential new hit for a gene associated with cognitive function and aging. So, but you can do, the point is that you can apply this type of approach for pretty much any problem. I mean, as I said, you can apply it to Game of Thrones characters. You can apply it to sociological problems. You can apply it to any disease or process for which you have, you need to just have a starting seed list of genes associated with that disease or process. That's the only thing. So there's a lot of applications for these machine learning approaches. Now, the last thing I'll mention, which draws upon what I started referring already, is that we've been I started as a model system person, and we've done a lot of model organisms. We've done some work in mice and rats as well that I don't have time to go into today. But really, one of the things we want to do, of course, is to find something that works in humans. So we've been doing a lot of work in trying to incorporate more human data into our analysis. As I mentioned, this collaboration with Ian Deary. We did this project called Longevity Map, a database of genetic association studies of longevity. We're using Framingham Heart Study data, UK Biobank data, applying machine learning approaches to discover new genes associated with aging phenotypes. One of the things we're trying to do also with King's College London is look into patients that take different drugs, the drugs that we defined in C. elegans, because some of them are used in the clinic, and look at their epigenetic clock if these patients are aging faster or slower. And just to give a couple of quick examples, we can also look into, for example, cell senescence in human tissues. That's something we've done. So, we have our signatures of cell senescence, and we can actually infer, as I said, looking at the literature, what these genes inhibit or promote cell senescence. And so we can go into human tissues, which you see here, and see if the gene's going up or down. And the most interesting thing is when you look at promoters of cell senescence, you see an overrepresentation of genes being overexpressed. So it doesn't happen in all tissues, but in more tissues than would be expected by chance, you have an overexpression of genes that promote cell senescence. So at least in some tissues, you do see signatures of cell senescence in vivo in humans. The last example I'll mention is a recent analysis that we published some months back, actually. This was led by Cassit, a brilliant PhD student in our lab, essentially looking at age association patterns using multiomics data across different cancer types. So basically, you have cancers of different ages, but characterizing changes in cancer using transcriptomics, genomics, epigenetic data, and how that changes with age. So, this was a very big analysis with collaborators in London as well that I don't have time to go into detail. I'll just mention two quick things. The first thing is that you see here there's an increase in mutations and genomic instability in tumors from older patients. It is not a perfect correlation, it's a big cloud, but you do see this increase in mutations and genomic instability in older ages. The other thing I thought was quite interesting was that I say regulated is an hypothesis, but there's a strong correlation between transcriptomic changes and DNA methylation changes. So possibly the transcriptomic changes are regulated by DNA methylation changes, so changes with age in tumors. So that's something I thought was quite interesting as well. Again, I don't have time to go into details, but it's an example. It's a very big work, and it's a good example of applying some of these methods, computational methods, to these human data sets, large human data sets, to gather new insights. So with that, let me just finish by summarizing. I told you that, well, you know that aging is a major challenge of medicine, of biomedical research, but it remains a mystery of biology. I think one of the issues we have in Phil, happy to discuss that later as well, is that we don't focus enough on trying to understand aging. I mean, there may be reasons for it when we can discuss it later, but there's a lot of focus on interventions and not so much on understanding aging. And I would argue we still don't understand the process of aging. We only have hypotheses so far. I thought of our GeneAge database, which over 2,000 genes, which really showcases how there is a digital code to aging up to a certain degree that modulates how we age and how long we live. And if we can crack that code, then that would be fantastic, and that would open the doors for interventions. So that's really, I think that's the rationale for my work since I started 20 years ago. And that's why there's this big focus of our lab in computational approaches, trying to decode that information, trying to understand how these very complex biological systems result in aging. I told you of our cell senescence, multidimensional systems analysis that reveal new candidates that we tested and validated experimentally. So reveal new genes associated with senescence. I told of our network pharmacology methods that showed new compounds and new drugs that extend lifespan. And I told you of our machine learning methods, which, as I said, can be applied to many different processes and diseases. And lastly, I told you about human data integration, which is something that we're trying increasingly to do, integrating human data. So going all the way from model systems into human beings to try to identify, to try to predict. Really, that's the goal, is to make biology predictable, trying to develop models that predict which genes are going to be more important, which compounds, which therapies are going to work. That's one of the big challenges we have, of course, in pharma, and that we're trying to address. So with that, I think my time is up. So thank you very much for your time and attention. I would just mention, reprints of all our papers are available. This is Cassit who did the analysis, Daniel, the cancer analysis. Daniel did the machine learning and some of the machine learning analysis. So thank you very much for your time and attention. Happy to take questions and discuss further.
Thanks for the talk. There's a lot of questions, so we'll try to get to as many as possible. The thing that comes up to me, you mentioned that aging is so plastic, and you've identified genes and compiled databases of genes that affect aging. What's your thinking on this? Why is it so plastic? When I started in the field, people said, single genes aren't really going to affect aging, even though we already had a couple at the time. And people were skeptical, and now it seems like every other gene you look at affects aging.
Yes, I think that's a great question. I think it is surprising. I think if you read some of the evolutionary biologists' works in the 80s, they would say there is not going to be a single gene for aging. I think there's a Linda Partridge paper in the 90s, like months before Andrej Bartke published his study, saying you're not going to find a single gene that extends lifespan in mice. And then Andrej Bartke months later published literally a single gene in mice that extends lifespan. I think so. On one hand, I think evolutionary biology is not predictable. I think you have to take a grain of salt what evolutionary biologists tell you. That's something Gary Ruvkun. Sorry.
You said it, not me. I just want that in record.
Well, Gary Ruvkun, I don't want to misquote him, but he said something along those lines. I think it's, on the other hand, I guess it makes sense for aging to be, polygenic, multifactorial, and that you are going to have individual genes that can have an impact. Keep in mind, of course, that in a lot of these interventions, like C. elegans springs to mind, you don't actually know what the worms are dying of. So, the worms may be living longer, but are they aging slower? I think in some cases they are. I think in mice, for example, the growth hormone IGF-1 manipulation, they are. But when you look at some of the pharmacological interventions, for instance, rapamycin springs to mind. It's very robust intervention. Are mice aging slower because they take rapamycin, or do they just have less cancer? and they die less of cancer. I think David Harrison has done a little bit of work, but I don't think that's been conclusively shown yet. So there's lots of ways you can make animals live less or more, and that's what we're discovering. But there's one thing I would point out, which is actually the most we can extend lifespan has not really changed in mammals. I mean, the most is caloric restriction, which was discovered in the 1930s. So, and that's about 50% life extension. So we haven't, I mean, there seems to be this barrier. I mean, you can extend lifespan by about 50% or up to 50%, but so far, not more than that. And I think that may be telling us something as well.
Yeah, that was my next question. Just briefly in defense of rapamycin, and along the same lines, Rich Miller came out and said, when they first published the data showing an extended lifespan in mice, that it's probably just preventing cancer. And then 6 months later, he published a paper showing it delays a whole range of age-related conditions and mortality through different mechanisms. So, yeah, I don't think it's all known yet, but I'm still hopeful for it. But coming back to your last point, which I think is interesting, you have 6 times in yeast, 10 times in worms, and 40% in mice in terms of lifespan extension by single gene manipulation. What does that say for humans? I mean, do you think that we're on the right path with these genes? Is that going to go down to 10%, or is it still going to be 40%? I mean, we don't know, but speculate a little bit for me.
Yes, no, that's the $1 million question. I mean, and there's a lot of companies on aging, and I think a lot of the reason we have this growth in biotech, longevity biotech, is because of these discoveries of how plastic aging is. But I'm not avoiding your question. So my guess would be that it's going to be less in humans. Because even mice, I guess one of the issues with model systems is that, there's 2 issues. One of them is that it's short-lived. So, maybe what makes Bernard Streller made this point decades ago is that maybe what makes a worm live longer, humans have already, right? So it's been fixed in humans already. The second point, of course, is that, or the second limitation of model systems is that most of them are genetically homogeneous strains. You see that in C. elegans, you see that not in all, like the ITP don't do that, but you see a lot of the studies in mice are done in genetically homogeneous mouse strains, typically black 6 mice, which is basically a fat mouse. So, if you're using a fat mouse to manipulate IGF-1 and insulin, it makes sense that it lives longer. But what happens if you take those same genetic manipulations and you put them in a small mouse or smaller that doesn't get obesity so much? That's a big question now. And when you do caloric restriction across strains of mice, as I'm sure you're aware, you don't get life extension in all of them. So my guess would be that, again, speculating, and I could be wrong, would be that when you look at the genes, if you could extrapolate the mouse genes to humans, I think some of them would work in some people, but possibly not in everyone. That's a big, and that's the big question as well. And that's why we need precision medicine and this predictive biology to try to predict. I mean, which genetics variants are going to be beneficial in which people in different genetic backgrounds. So that would be my guess. Some genes will work in some people.
There are a lot of black 6 related humans out there too that are overeating and have it may be a good model for some people, but I agree. And already some of our data with AKG as an intervention suggests that it works better in people that are not aging well compared to the ones that are. And so, I don't know if it'll be that will be the case with every intervention, or that's just a unique finding, but certainly I don't think anything is going to work in everybody. And that brings us to this precision you're mentioning.
But that's I think one problem. I mean, some people have pointed out, which is that, okay, maybe in these model systems like fat mice and so on, unhealthy animals, we're just, bringing them to normal, right? Yeah. So, okay, so maybe caloric restriction or some of these manipulations or rapamycin will work in individuals that have unhealthy diets or unhealthy lifestyles. But for the person that already has a healthy diet and has a healthy lifestyle, they will not work And I think that could be a problem, I would say.
Yeah, we don't know. You mentioned a lot about we need more mechanistic understanding, and I certainly agree with that. And I think that as somebody who was trying to find genes that affect aging, mostly in yeast in my case, but in other species as well, our hope was we could take those genetic mutants and get to mechanism. But it's proven very difficult, and the classic approach has been epistasis, where you knock out a gene that you think is downstream and see if the effect of your gene goes away. But every time we do that, we get a complex, network of gene interactions, and it's never just simple linear pathways that point to causality and resolution of aging pathways. And so, I mean, our genetics, what do you think about that? Or is this genetic approach, it's telling us how to do pharmacology, but it's probably not telling us as much about mechanism as we'd hoped, I think.
I mean, you touched on the big problem. I think biology is just too complex for us to understand. I mean, I would say, the example I usually use is Alzheimer's disease, because Alzheimer's it's just one tissue affected. You would think it's simpler than aging. You would think Alzheimer's is part of aging, and so it's just one component. And there's more scientists, there's more funding for Alzheimer's. I think it's like $3 billion every year that they get funding. So it's an Altos Labs every year that they get, and there's not a single drug. And I don't think we can say we understand the mechanism of Alzheimer's, although there are hypotheses. So I think the problem is that biology, in particular of these complex processes and diseases, is really, really complicated. And when you start trying to do digging about it, you just come up, okay, well, that's this gene network, and then what does that tell us? And we're still not able to understand it. Now, I think human biology is still very complicated. But so, for example, when you focus on cell senescence, that's a simpler phenotype than organismal aging. And so I'm hoping, well, at least some of the mechanisms of cell senescence are understood, like telomere shortening. So I think we have to take it one step at a time. I do think we should do more mechanistic studies, although that sounds good, but as you just pointed out, it's really, really hard thing to do. And in fact, I would add, one of the things I've noticed over the years is that you have all of these biotech companies working on aging, but then most of them pivot. They pivot away from aging into age-related diseases. Why? Because they go, oh, this is really, really complicated. We're not going to crack it. We might as well do cancer. Most of them, not all of them, pivot to cancer, but quite a few pivot to cancer or another age-related disease, or some sort of observational study that doesn't really tell you about the mechanisms, because it's really, really complicated. And I think that goes back to the complexity of human biology. We're still a long way to go, I'm afraid, until we understand human biology.
Well, we could go on forever. Maybe I'll bring Diogo in to ask some questions. And actually, no discussions in Portuguese, guys, huh? What'd you say?
Well, was that too pessimistic? I don't like to sound too pessimistic, because I think we will crack the code eventually.
You're giving us the glass half empty comments here, I'm telling you.
So it's going to be my pleasure now to start the Q&A, Pedro. And it's a pleasure to be here. Actually, I'm on vacation, but I took the time to have this honor. We have a lot of questions, so let me try to summarize them. So one of them is, what are actually the computational models that your lab is using to study the causal relationships, so not correlation, between certain genes biomarkers and health outcomes?
So that's a good question. So first of all, computation approaches give you predictions. They don't give you certainties, right? So you can use, for example, in co-expression analysis, you can do something like directional co-expression analysis. You can actually tell if it's going for, well, if you have sufficient data, you can tell if a particular pair of genes, if they're, it's a gene A regulating gene B or vice versa. So, and you can bring in transcriptional regulation data for that as well. So in some cases, you can infer causality, or you can predict causality. Let me rephrase that. But it doesn't work all the time. And there are cases where you have a big network and you have, so this gene seems to be causal here, but then you have another gene that is causal there. So So it's not always easy. So I would say you can use these methods like directional co-expression analysis. You can look at transcription factor binding sites, or you can incorporate data on transcriptional regulation. You can infer causation in some cases, but it's still not, it's still not 100% accurate. That of course is the problem. So it allows you to generate an hypothesis about causation that you can test experimentally.
Okay, great. Yeah, exactly. We also have a question from Camille Pavis, and I actually remember we having this conversation years ago when I was still in your lab, and it's about the short-lived species, basically, or intervention. So GeneAge contains genes that are life extending and lifespan shortening when knocked out or overexpressed. However, Camille is a bit worried about the data coming from the study showing that life shortening because it's easy to harm an organism without affecting aging rate. So have you considered this bias?
I'm not sure so I agree. GeneAge, I guess, will reflect biases in the field. So GeneAge is taken from the literature, and so it will reflect the biases that you see in the literature, that in the way scientists do experiments. I think there are biases. Scientists tend to publish positive results more than they publish negative results, and that's a bias. And I'm sure GeneAge has that. So I guess the point is that there are biases. I'm not entirely sure what you mean by addressing. I'm not entirely sure how to address it. That's the problem. It is what it is, basically. So we're aware of them, but again, it's a reflection of the biases of the scientific community, so to speak. I published a paper earlier this year which is that how every gene is associated with cancer.
Yes.
And that reflects biases because most people who work on any gene will try to associate it with cancer. It doesn't mean that every gene is causal in cancer. It just means that I think it reflects, again, biases from researchers in studying cancer more than any other process. So not just GeneAge, but the scientific literature is unfortunately full of these biases. It's not that easy to address, however. I guess it depends on the kind of analysis you want to do. There might be computational methods to address. We've done, in our 2016 paper, we corrected for publication biases. So correcting in the networks for the number of publications associated with each gene. You can do that. But I think it's impossible to correct for all of them. So that's the answer to the question. Because I think it will depend on how you go about it.
Yeah, I think so. And I think the other level, so for this question is basically, if we repair a short-lived animal model organism due to some kind of intervention, are we actually gaining some insight to the natural aging process? Or only if we work with long-lived species and extending lifespan?
That's a very good question. And again, I think, and that's something that people have pointed out before. It may be, maybe, as you say, maybe they will not be relevant to long-lived organisms. It's possible. I agree, but I don't know for sure.
Okay, thank you. So there are also more questions. I think it's related with, I suspect, your Multiomics study that you presented, and basically it says that if then in the end, it's the post-translational modifications of protein, like DNA methylation, ubiquination, etc., that are drivers of cell senescence, that mark the drivers of cell senescence. Is this true? So basically, I think what they're trying to say is it an egg and chicken problem? So is it genomic damage that somehow dysregulates the epigenetics, or is it the epigenetics? What can you see from data that somehow dysregulates how the genome is expressed and translated?
Yeah, so that's an interesting question. Something we've been discussing actually. I think there's different lines of thought. Frankly, I don't think the data and the analysis we've done tell you for sure one way or the other. You have correlations, and you have, how can I say, you have associations, you have signals from epigenetics, from DNA repair pathways, and so on. But yeah, what is driving what? I don't think we can make a strong assumption. My more broadly speaking now, I mean, my broad take is that I am not fully persuaded, for example, about the role of mutations in aging. I think that DNA damage may play a role in aging in some way. I mean, David Sinclair has this concept that you lose epigenetic information because of DNA damage. That may be the case. Maybe there's loss of epigenetic information anyway without DNA damage. That may be the case as well. And I think, for example, the Horvath clock shows that you do have epigenetic changes from very early ages, from embryos. Is that being driven by DNA damage? You do have mutations and you do have damage even in early stages of development, but I wouldn't necessarily assume that's driving aging. So I think there's still a lot of open questions. So I think it's a great question. I'm afraid I don't really have an answer, but yeah, I think there's different ways of thinking about it. One is that you have DNA damage that results in epigenetic damage or epigenetic information loss that drives aging. There's the concept that you have DNA damage and that drives aging by itself, or mutations. Or maybe it's just epigenetic information loss by itself that drives aging. I think all of those hypotheses are out there. I think they each of them could be right, but it can also all be wrong. Maybe something else. I'm not sure.
How persuaded are you by cell senescence, since we're on the topic?
Not very much. So I mean, I did my PhD. Look, I did my PhD in cell senescence, and then I didn't work on cell senescence for 10 years, maybe 8 years. So I think that should tell you something about how I think cell senescence is important. Now, they had the papers from Jan van Dersen's lab on cell senescence that made me go, okay, so maybe cell senescence does play some role in aging. So that made me think that could be a possibility. But I would say the jury's still out, and I'm not that convinced about cell senescence as a causal factor in aging. I see cell senescence as it's a stress response. So, it's like the fire department of the body. You get something wrong, you get cell senescence because it triggers inflammation because it wants it to repair or it wants to fight infection, whatever. Something's wrong, you have cell senescence. So you have a correlation. And of course, if you have too much of it, it will be bad. Just like if you have too much inflammation, if it's bad. But if you don't have inflammation, you can't live either, right? And if you don't have cell senescence, it's not good for you either. So, there's like a Goldilocks zone for cell senescence. But whether it's causal in aging, I would say I'm not convinced, but I also keep an open mind. I think you have to be humble about biology and about, maybe there's things we don't know, and there's unknowns, things you don't even know you don't know.
So maybe I should continue with some more questions. So there is another one that is related to basically what are the major obstacles and limitations of applying, these computational methods that you specialized on?
So the computational methods are not, it's not that difficult thing to do. I think there are limitations. For example, sometimes you have data limitations. You don't, we can't, so for example, single cell sequencing, you have loads of data from mice nowadays with aging coming up, from Calico and from others. In humans, it's a lot more complicated. I mean, the brain. I mean, people for some reason don't seem to be wanting to give brain samples for research, go figure. So it's we have limitations getting quality data sets, particularly from humans. That's a limitation, not just in aging, but in aging as well, because you need samples from older individuals, which is not always easy to obtain. So you have limitations sometimes in terms of data. And for this kind of computational approaches, you need quality, you need quantity, and you need quality data as well.
So in your presentation, you actually said that there is now a shift from data quantity being the limiting factor or generating data to be able to, the data analysis becomes now the major focus. So what are the limitations in there?
So, absolutely, that's I guess in a slightly different context. What I mean is, it's easy for you to sequence your genome. It's cheap and easy. You can do it fairly rapidly in a matter of days, and you get a load of data. So in days, you can get your genome. But then it will take you weeks, if not months, to figure out really, what it all means. So that's what I mean, is that it's very easy to generate data. The analysis, it's more complicated. I guess what I'm saying is that more broadly in applying machine learning or computational analysis, sometimes data quality will be, and quantity will be a limitation, particularly human data. So, for example, mouse data, I think there's plenty of it. So those are some of the limitations, I would say, because if you look at other fields, like cancer, for example, there's way more data. So that would be one limitation. I guess it's not a computational limitation, but I think that the biggest bottleneck in aging research is really the time it takes to do experiments. And that's not something you can change with money or technology. If you want to study aging, even in a mouse, it's going to take you years. You can have all the money from Altos and Calico and Google, all the money in the world. It's not going to change that. You still have to take some years to do those experiments. And if you want to test something in human beings, for example, if you want to do a clinical trial, that's still going to take time. That, again, there's no way around it. There's no way technology or money can solve those problems. And so I think those are intrinsic limitations of studying aging that other fields like cancer don't have. That are hindering progress in the fields. But again, they don't really have an easy solution. What I hope some of computational work, in silico work we and others do, is that we can reduce the number of experiments. So instead of doing 10 clinical trials to get one success story, you only need to do 3 or 5 clinical trials. Or you do 10 clinical trials, but you have 2 success stories. So that's what I hope to change, is to make the process more efficient, so you get more hits for the same amount of experimental work you do or clinical trials.
So I think I have to jump in there. We're running out of time. Thanks a lot for joining the show. We'll have you back on sometime, and look forward to seeing you around soon.
My pleasure.
Anytime. I want to remind everyone to use the chat function and the panelists and all attendees option to leave comments and feedback on the show. Look for news from our Center for Healthy Longevity, on the School of Medicine, and also on the School of Medicine in the end credits. Also, you can scan the QR code and get into our Twitter account to continue the discussion on aging. And we can also try to link that our Twitter account to some of the web pages that Pedro mentioned, as well as his Twitter account, in case you're interested. We've recently launched our website for the Center for Healthy Longevity as well, under NUHS. And so there is a link for you where you can go visit that website and look at what's going on if you choose to. Next week, we have Robin Daly, who hasn't given us his title yet, but it's probably going to be related to exercise and aging. I'm going to go out on a limb and guess that. I'll be doing the host, so that'll be an interesting conversation. And I want to leave you with our final video, which is about an 86-year-old Agong.
The thing about dementia is that you're in your own world. So you only remember what you want to remember and what you like. Agong likes horse riding, so Agong will always remember horse riding. My name is Andre. I am Agong's grandson. I'm mainly in charge of handling Agong, bringing him to places, making sure he does things. Because Agong can be a bit of a handful. Agong is very serious towards horse riding. If we tell him that he cannot go for horse riding, I'm very sure Agong will figure out a way to leave the house and then go for his horse riding by himself. Agong gave up his favorite snacks, spend most of his time exercising, climb up and down the stairs, do squats, because the stables required him to have a clean bill of health, and he also had to be under a certain weight.
每天上下中午到傍晚6点，因为骑马必须有脚力，在马上才能站起来。
开心吗？
开心，因为一生一世很想骑马，但是没有机会。现在有机会了，但是尽量的学啊。福建话说。 假到了，我更老了。
以后 后会跑比较快吗？
跑快现在还不可以跑，心想是要跑，可是要等脚力有了才可以跑。说到骑马，什么人都可以骑，只要他肯学肯练，就会骑。
A lot of us don't have is the patience which Agong has. He will make time to make sure the horse eat without showing any sign of like I'm tired. Before I met Agong, I thought about maybe 60, my horse riding, everything is going to be over. But now, I'm thinking that till my very last day, I will have to ride horses. That's the kind of inspiration Agong has put onto me, and he has told me age is just a number.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.